Assalamualaikum warahmatullahi wabarakatuh and good day. I'm Anissa Maisara binti Aziz, S56441, and I will be presenting my final year project titled Review on Environmental DNA, DNA Analysis for Biomass Assessment in Fish and Aquatic Organisms with the supervision of Dr. Nur Asma binti Arifin. DNA analysis can detect DNA traces in a cellular or extracellular frame in feces, secreted mucus layers, gametes, and skin cells. It can also be used to determine the presence or absence of species, as well as their abundance or biomass. Since absolute abundance estimation and conventional approaches are often time-consuming and expensive, they are not included in standard monitoring. For overcoming technological difficulties in collecting data on fish stocks, DNA analysis could be a game changer. What is the problem? The problem is the fishery independent surveys are often costly and logistically demanding compared to eDNA analysis. Commercial catches are becoming less of a data source for evaluation of biomass due to decreasing yields and increased competition. Multiple biases such as variable fishing effort or efficiency and/or hyperstability are present in data from fishery-dependent methods. The significance of this study is that eDNA can be collected and used to determine the presence and distribution of a species at a lesser cost compared to traditional survey methods, and eDNA can be a general fisheries technique suitable for rare species and managed stocks in freshwater and marine environments that does not harm protected areas or the species that live there. eDNA monitoring can cover vast areas and gain access to water environment that are inaccessible to traditional equipment. The objective of this study is to determine the current status of eDNA analysis for biomass assessment in fish and aquatic organisms. What is DNA? DNA, or deoxyribonucleic acid, is a macromolecule comprising of all living bodies' genetic material. It preserves and transmits the genetic features of all recognized living organisms. According to Marshall 2007, DNA has become the universal tool for preserving the memory of life. What is eDNA? eDNA, or environmental DNA, was first used in 1987 to describe a method for extracting microbial DNA from sediments, but it was only in the early 2000s that the term became common, mainly among microbiologists. It is a DNA that can be extracted from natural samples without capturing target organisms. It is characterized by a complex mixture of genomic DNA from a variety of species. It can be found in both cellular and extracellular forms, and it can come from a variety of sources. The first step in eDNA methods is sample selection, with grab sampling and composite sampling being used in the current water sampling strategy. The second step is eDNA capture. The three main methods for extracting eDNA from aquatic environments are filtration, precipitation, and centrifugation. The third step is extraction or purification stage, where extraction kits are used. And lastly is the qualitative and quantitative analysis of eDNA. Conventional PCR approaches may be used to investigate the presence or absence of fish in a given area. In eDNA research, real-time qPCR has become the standard tool for species-specific detection. This is the schematic of eDNA workflow for samples collected from ponds by Harper et al. 2018. This DNA analysis can increase the geographical and temporal coverage of population evaluation and monitoring while being less invasive, time-consuming, and costly. It could also speed up sample collection, lower data collection costs, and prevent negative effects for the species under analysis. It is expected to become a valuable management method in ecology and management, and it has great potential for assisting in the resolution of technical problems associated with sampling in remote areas. These figures show a positive relationship between the concentration of eDNA from species studied and their biomass and abundance. Estimating the biomass of fish populations is generally accomplished by collecting in situ fishery-dependent or independent data as a foundation for stock evaluations. Typically, catch data from vessel logbooks or scientific sample aboard or at landings are used in fishery-dependent techniques. Due to decreasing yields, commercial catches are also becoming a less reliable data source for evaluation in certain industrialized countries. Genetic studies using eDNA samples are one alternative to traditional survey methods. Mucus, feces, urine, gametes, and skin cells are released by all organisms, resulting in intracellular and extracellular DNA being suspended in the water column and detectable for up to 60 days in aquatic species. This study will be done by conducting a search on Web of Science, Google Scholar, Scopus, and Wiley Online Library for peer-reviewed articles relating to environmental DNA with keywords such as eDNA, biomass, fisheries, marine, freshwater, and aquatic from the year 2000 to 2021 from countries all over the world. The species, the objective, the methods, the location, and the year of the study will all be searched for the data. This data will be collected, tabulated, and partitioned by the researcher, study site, and species study. The study is expected to determine the current status of eDNA analysis for biomass assessment in fish and aquatic organisms, where it will provide more verification on how eDNA concentrations correlate with biomass of fish or aquatic species. This research will also look at how eDNA is a better tool for assessing biomass compared to traditional survey methods. The finding of this review will also help local researchers in better understanding how eDNA analysis works and how it can be used to evaluate local fish biomass with less time, expense, and bias. These are the main references referred in this study. And last but not least, I would like to thank my coordinator, supervisor, family, and friends.